Good morning, guys. Start coming for lecture, guys. It starts already. Guys, it starts already. Good morning, please. Guys, the lecture starts already. Good morning. Guys, good morning. Good morning. Hi, my dear. Good morning. Nice to see you again. And I hope you are doing well. And those who are infected with corona, I hope you don't have severe symptoms because I hope you mostly are vaccinated. So it will, even if you are infected, will go not so severe. So, and today's topic is not about coronavirus yet, but coronavirus lecture is coming up. So today we're supposed to discuss the RNA viruses as causative agents of the respiratory infections. And of course, coronavirus is also RNA virus that causes respiratory infection. But as I said, we will have a separate lecture for it a bit later on. So today we'll discuss the very well known, very old pathogens that belong to two groups, Paramyxoviruses and Orthomyxoviruses. The term myxo or myxoviruses denotes a large group of enveloped viruses that are able to attach by their glycoproteins to cell surface receptors. So, based on the fundamental features of these viruses, we usually distinguish two large groups. These are Orthomyxoviruses and Paramyxoviruses. And so you will have a class for these both Orthomyxoviruses and Paramyxoviruses. So we start with the families Paramyxoviridae and Paramyxoviruses. Several times the classification of this family had been rearranged. By now, we distinguish several or at least two subfamilies, Paramyxoviridae here, right, and Pneumoviridae. And within this, we have several genera. And these genera are Paramyxoviruses with 4 serotypes, but these 4 serotypes are distributed within 2 genera. In Paramyxoviruses, we have human parainfluenza virus type 1 and 3, and interestingly, that human parainfluenza viruses type 2 and 4 with subtypes are placed into a different genus, which is rubella virus, together with the mumps virus. This is, my dears, unusual, but that's what DNA says, right? So, then one more genus is morbillivirus with measles virus, along this solo in this genus. And then we have comparatively new viruses, and there's a separate genus had been arranged for them, Henipavirus genus, and there we have Hendra virus and Nipah virus. These two are comparatively new. You can read a bit about them in the internet, but that's not of very high medical importance yet. Maybe in few years they also will cause some epidemics, though we put an eye on them. And the last virus in the family Paramyxoviridae is so-called subfamily Pneumovirinae and the genus Pneumovirus, that is respiratory syncytial virus. That is the typical virus for the whole family. So, what should we know about the basic characteristic? These are enveloped viruses. You can see here the nucleocapsid here and the envelope with the surface glycoproteins, and inside it has negative sense single-stranded RNA molecule. The morphology is pleomorphic, or most commonly they are spherical. And you can see here that this type of the symmetry of nucleocapsid symmetry is helical. So nucleocapsid follows the structure of the supercoiled RNA molecule. So as we take a look at the proteins that are found in the virion. So inside we have nucleic acid. As we said, there's a RNA with the replicating machinery. And then it is covered with the matrix protein. And then we have an envelope with the attachment proteins. But then, my dears, every single Paramyxovirus has two surface proteins, glycoproteins. But then the one is common for all Paramyxoviruses. This is so-called fusion protein. Take a look. This is shown here. So it is same for all, as we said here, for all Paramyxoviruses. But then the attachment proteins can be different. And regarding the presence of this attachment protein, we may classify them into three large groups. One type, usually this pneumovirus, has just G protein. We call it G protein or glycoprotein. It mediates the attachment. Then measles virus, as you see here, measles virus and parainfluenza type. So they have HN virus. And mumps virus has H virus for attachment. So they are different. And also they have very minor so-called SH protein that can inhibit tumor necrosis factor alpha signaling pathway. So that's one of the ways how they may escape from the immunity. So here you can see the genome of the virus. And in the genome, you can see that several types of the proteins are encoded. So the L protein, that's supposed to be somewhere here, right? See, this is the large protein. And the function of the large protein is to make the transcriptional complex to produce mRNA from negative sense RNA. So, but then also we have the very small phosphoprotein. See, this is the one. And the phosphoprotein is encoded by this part of the genome. The phosphoprotein make a complex with the large protein, and these two proteins form so-called RNA-dependent RNA polymerase that mediates the transcription of minus sense RNA to positive sense RNA. Then here we have M protein. We forgot N protein. N protein is the nucleocapsid protein. here that follows the subunits of these proteins or capsomers. They follow the structure of the RNA. So then, as we said, we have M protein. M protein lines from inside the envelope. And as it is known, we believe that the M protein mediates the morphogenesis of the virus, so we will touch its function when we discuss replication a little bit. So this is a matrix protein. See, it is located somewhere in between the nucleocapsid proteins, the large proteins, or the complex, how do we call it? Transcriptional complex, and the surface glycoprotein. So we have lipid bilayer, and I would like to remind you that in the complex viruses or enveloped viruses, the lipid bilayer is taken from the previous host cell membrane. So this is not virus specific. Sometimes even some cellular markers may stay on the surface of this cell membrane. But now this cell membrane is modified with viral glycoproteins. And you can see here the fusion protein is this one that is common in all Paramyxoviruses, and the hemagglutinin protein or some variants of the binding proteins that is here, right? So because it can be hemagglutinin, then it can be HA, hemagglutinin with neuraminidase activity, and also it can be glycoprotein. So, and here we can see we just briefly mentioned the replicative cycle of these viruses. But my dears, because they are absolutely typical for minor sense RNA viruses, and because they have a single piece as a genome, so they don't undergo very many antigenic variations. So the process of new strains development is it doesn't really occur at the very high rate. Okay, so when we take a look at the replicative cycle, you see that the virus may attach to the host cell receptor with the surface glycoproteins, so H, N, H, or G, and then enter through the fusion of the viral membrane with the cellular membrane with the help of fusion peptide or fusion glycoprotein. F stands for fusion, so that's the way how these viruses, all of them, enter the host cell. So as soon as nucleocapsid enters the cytosol, and the envelope will be lost on the surface. So then the RNA-dependent RNA polymerase complex made up of two proteins, polymerase, large protein, and phosphoprotein. They start producing mRNAs using minor sense RNA viral genome as the template. Then the process of viral protein translation occurs. So, and the capsid proteins will be produced. Then the some regulatory proteins will be produced, and surface glycoproteins also will be produced. So then, as a result of this, the nucleocapsids first will be developed, and then during the replication of the viral RNA, so the viral genome will be transcribed into long mRNA, and then the mRNA using as a template will be transcribed into progeny RNA genomes, many minor sense progeny genomes. So then maturation and assembly of the capsid occurs. But then M protein will be transported to the inner side of the cell membrane, of the cellular membrane, and then the ready nucleocapsids will be taken towards the modified cell membrane. Then the process of budding occurs. So where the surface glycoproteins, so because they will be expressed on the surface of the infected cell, and then the process of budding as a result will yield in the releasing of the newly synthesized, newly replicated viruses. So this is a process of the Paramyxoviruses replication. We won't discuss them. There's nothing really very difficult in understanding of these viruses. But we would take a look at the measles mainly, because this is a very common, highly contagious disease that used to cause epidemics, and we stopped these epidemics just with the help of vaccination. So, but then few words about the disease itself. So, and how viruses causes this disease. So, measles virus infection usually starts as a respiratory illness. But then this respiratory illness usually is complicated with the immunosuppression. And then because virus also may infect brains, so it may have a very severe consequences. So that's why we try hard to eliminate the measles from the human population. By now we just stop the epidemics. We didn't eradicate it completely, but we tried mainly because of the very severe complications it may give. So you have to remember that measles is neurotropic virus, and the complications it may cause is, for example, acute encephalitis. This is the direct complication of acute measles virus infection. But apart from acute encephalitis, they may cause very slow progressing chronic disease of the brains that is called subacute sclerosing panencephalitis, SSPE. I hope you understand what is it, panencephalitis, like Pan American. The flights through the whole American, panencephalitis, when all regions of the brains are infected. So then here some receptors are shown that supposed to be used by measles virus to enter the cells, right? So, and this is the so-called SLAM receptors on the surface of the dendritic cells. So it means the dendritic cells can be infected with this virus. And this is also the receptors that are found on the surface of the T CD4 positive lymphocytes that also may be recognized as a very good target for the measles virus. So then this receptor that is called MVR receptor is located on the surface of the brain tissues. And CD46, this is the receptor on the epithelial cells, and so do the CD150, that is the one. So usually measles virus enters, that's the pathogenesis of the disease, enters through the upper respiratory tract by using SLAM or signaling lymphocyte activating molecule. That's the definition for abbreviation SLAM. So in upper respiratory tract, measles virus use them as a receptor. So then the measles virus infected lymphocytes usually enter the bloodstream. Please pay attention that measles virus don't replicate very much in the mucosa layer at the beginning or initially. So it rather infects the dendritic cells or T cells. Then measles virus infected lymphocytes enter the bloodstream, and measles virus replicate in lymphoid organs throughout the body in lymph nodes and the accumulation of lymphoid tissues in appendix and so on. So then the measles virus immune cells transport measles virus to epithelial cells in various organs. So in the airways, in the intestinal tract, in the bladder. So in fact, they spread the virus along the body, including lungs. And then when measles virus replicate abundantly in the epithelial cells, the measles virus will be actively released into the airways, and respiratory aerosol containing measles virus particles will be released, coughed out. So, and then that's when the patients start to be highly contagious. So consequently, respiratory aerosol of patients contain very large amount of measles virus particles. So it is a virus with one of the highest epidemic index. So then, the result of the common result of the measles virus is immunosuppression. It is a major cause of infant deaths associated with acute measles, and therefore of substantial clinical importance. Major hallmarks of this generalized modulation of immune functions are lymphopenia, that is very difficult, that result from the depletion of the measles virus infected T lymphocytes. And this depletion may occur, so it appears that measles virus may infect T cells on any stage of lymphocyte development, and evidently expression of the major measles virus receptor CD150 plays an important role in the targeting of that particular cells. Then, apart from direct killing of the T lymphocytes, a prolonged cytokine imbalance consistent with suppression of cellular immunity to secondary infection, and also silencing of peripheral blood lymphocytes which cannot expand in response to ex vivo stimulation. What does it mean for those, and for especially those who don't study immunology? If we have the peripheral resting lymphocytes, they don't recognize the antigen yet, so they're supposed to be to start proliferating when they recognize an antigen. But the measles virus make them silent, not possible to respond to new stimulation with the unknown virus. So that is the immunosuppression regarding the future infection. So they will not be able to react towards future infections. That's very bad. So then we come up to the very common complication. It is quite rare but well recognized as a result of previous acute measles virus infection that is called subacute sclerosing panencephalitis. And here we can see three potential routes of entry of measles virus into the brains. Since the virus can infect neurons, it has then access to the brains via nerve bundles, right? So this is the in the olfactory bulb, and that may occur directly as a result of measles virus infection through nasopharynx. So as an alternative, the virus can replicate in the endothelial cells of blood vessels of the capillaries, right? And when the measles viruses are released there, they may be taken directly, or here, second ones, through the capillaries into the capillaries of the endothelial cells. And there they may release the viral particles into the brains of the patients, and so then the infection, the third way, the infection of monocytes, where they won't be killed properly, they can be transmigrate through the blood-brain barrier, so they enter the brains by the third route. So all three routes we believe may work. So now you can see the development of the symptoms in the measles. You can see that when the virus entered the body through air droplets, virus goes around the body and multiplies finally in the respiratory system. Then when it multiplies, the complex spots begin to appear. This is as grain of salts in the nasopharynx. Then these appear before the infected. So then as a result, after several days, the person starts to be contagious. And then only the catarrhal period occurs, where the catarrhal symptoms will be seen. Then the rashes begin to appear, and the rashes are rather the reaction of the immune system towards the measles virus than the activity of measles virus directly, because this is not cytolytic virus, so it doesn't cause the death of the cells where it propagates. So the cell-mediated immunity, T cytotoxic lymphocytes that kill the infected T lymphocytes are or endothelial cells of blood vessels are the major cause of rash development. And approximately on the day 14, the rash will appear, and around that time, the infected person stops to be contagious. And then rash fades, so then we have the possible persistency of the measles virus in the brains. And while they persist there, they don't cause very symptoms development. So until the virus infect and accumulate in many cells in all regions of the body, of the brains. And then the multiplication of the virus may cause demyelination of the brains. So then inflammatory cells will become in there, and immunopathological immune response will cause the damage to the cells. So it may lead to very fast symptoms development and usually death. So you can see here the virus replication, weight multiplies, so respiratory tract, local lymph nodes, blood, spleen, then thymus, liver, skin finally, right? And how it coincides with the development of the symptoms. First, temperature, cough, fever, conjunctivitis, and complex spots, and then rash, this is a catarrhal period, and then rash, that is the result of the immune response, my dears. See, the period of rash coincide with the rise in the immunoglobulin development and the production of or activation of CD8 T lymphocytes. See, these immunoglobulins, and these are T lymphocytes. And as a result of this, we have immunosuppression. And at the beginning, we have a release of gamma interferon, but then it fades, and the immunosuppressive interleukins IL-4, 10, and 13, and pro-inflammatory interleukins will be seen in the much higher concentration than at the beginning of the process. So how do we perform the diagnosis of measles? Truly, usually we base our differential diagnosis on a clinical syndromes or a syndrome that is present, and this is a high fever, cough, coryza, conjunctivitis, and photophobia, then complex spots appearance, and then later on typical exanthems. So usually if we see the clear clinical symptoms of measles infection, we don't need any laboratory diagnosis. But then usually if we have the complications, for example, subacute sclerosing panencephalitis, then the testing may include anti-measles IgM or IgG antibodies. So usually IgG is paired sera. Then isolation of the measles virus is possible. And also the identification of measles RNA by PCR test is also possible. Quite often we may use histologic evaluation of skin lesions or respiratory secretions because they have very typical CPE, guys. This is cytopathic effect of the virus that is seen as giant cells, multinucleated giant cells formation or syncytial formation. Here, for example, you can see the syncytial keratinocyte giant cells in the skin. So that's because I would like to tell you, that's because I would like to underline. See, when we have a cell infected with the virus and virus replicates inside the cell, before the newly synthesized virions will be released from the host cell, the viral glycoproteins are expressed on the surface of the infected cell membrane. Then it appears that this cell may act as a virus partly, right? Because it has on the surface fusion peptide and the receptor binding protein. So it means that infected cell may bind with non-infected cell and fuse with it. So it means that the newly synthesized nucleocapsids can be transported from one cell to another cell by so-called cell-to-cell transmission without coming out of the cell. So, and that's important because when the virus comes out of the cell, it can be reached by antibodies, neutralized by antibodies. But the antibodies cannot reach the virus while it's inside the cell. So to eliminate the measles virus from the body, the cell-mediated immunity is crucial. Why? Because it may eliminate the virus-infected cells where the virus replicates. So neutralizing antibodies will be definitely not enough. So now a few words about how to stop this prevention and the control of this disease. My dears, we don't have any antiviral drug against measles. Nothing, really. So the only vaccination may help. And here we can demonstrate the morbidity of the measles virus. This is the cases per year. Since 1950 they were very high, and then you can see the dramatic drop of the cases number. Why? Because in 1963 the measles vaccine had been introduced. So, and that is the cases, and these are the deaths. So the both morbidity and mortality drops dramatically, but you see that from time to time we have some outbreaks. And not so far ago, the outbreak happened in the United States, in Great Britain, and some other countries. Why? Because they stopped the program of the vaccination, and some communities try not to, usually regarding the religious traditions, try not to vaccinate their children with a live measles vaccine. The problem is that vaccine used everywhere within the worldwide, right? This is the live vaccine. So that is the problem in some countries, and the only vaccination may help to keep the morbidity and mortality of the measles disease very low, like we have right now. I think that's enough for measles. Other infections is not very difficult to understand, so we shift to influenza virus right now, and we go on with the influenza virus. So here you can see some information about previous influenza virus epidemics. So that is the problem of influenza virus, that from time to time it causes epidemics and so-called pandemics. I hope you understand the difference between epidemic and pandemic. Just a scale of the infection, right? So, and the thing is that if it spreads through one continent, it is epidemic. And if it spreads via the continents, within at least two continents, that is appeared as pandemic. So the first very well recorded and described pandemic was so-called Spanish flu that occurred in 1918 within the World War the First, and it was H1N1 type subtype. And during this pandemic, around 50 million people died. And only in the United States they give this statistic. More than 500,000 people died. So, but then it was not really Spanish flu. Spain just claimed the number of victims of this flu. Of course, it happens in many other countries, but they keep it as a secret because that was the World War First. So then the second large pandemic occurs in 1957-58, and it had been called Asian flu, and the formula of the virus was H2N2. And first, this virus had been identified in Guizhou. It spreads to Singapore, Hong Kong, and then to the United States, and estimates worldwide deaths ranging from 1 million to 4 million. So a pandemic was predicted because that time it was already possible to monitor the appearance of new strains, and it was quite quickly identified. So, and vaccine against this infection was available in limited supply by August 1957. Then the next one occurred in 1968. It was Hong Kong flu, H3N2. The number of deaths between September 1968 and March 1969 was 33.8 million thousand patients, making it the mildest flu pandemic in the 20th century. But then we have repeated or return of the same virus in 1970 and in 1972. And in 1977, it was so-called Russian flu of H1N1 structure. A juvenile, it was called. Why? It was age-restricted pandemic. Why it occurs in young people? Because they if you take a look here, they didn't have the newborns. So the old people had some antibodies arise from Spanish flu pandemic in 1918. So, and in 1977, the young people, they didn't have any antibodies. This strain had been absolutely unfamiliar for them. So that's why it had been called juvenile pandemic. So then in 1997, we have first documented infection of humans by avian influenza A virus H5N1. Before we believed that happened, but there were no really evidence of this possibility. But then since 1997, very often the outbreaks of avian influenza viruses start to be registered that may occur in poultry, and also a limited number of human infections occurred together with epidemics in poultry. But the thing is that mortality rate in humans was very high at this initial avian flu infections. But then already in the 21st century, in April 2009, the very large outbreak and then pandemic occurs with a new strain, H1N1, that had been called swine flu. It began in Mexico. And in June, the World WHO raised its pandemic level to the highest level, phase 6, indicating widespread community transmission of at least 2 continents. But then we know that it spreads to all continents in the world. So now a few words about the virus itself. So it is a negative sense RNA virus, single-stranded negative sense RNA is segmented, and that is very important characteristic of the influenza virus that gives rise this feature gives rise to the high antigenic variability of this virus. It is an enveloped virus with pleomorphic nature. The shape of the virion usually spherical, of middle size, 80 to 120 nanometers in diameter. So, and it is covered with the envelope with two types of glycoproteins, hemagglutinin, HA, and neuraminidase, NA, or N. The problem is that the worldwide epidemics can be caused by influenza virus. So, and we believe it is a major determinant of morbidity and mortality caused by respiratory illnesses each year. And the influenza virus infections have been responsible for millions of deaths worldwide. And again, genotypic variations such as mutability and high frequency of genetic reassortment are characteristics of Orthomyxoviruses because of their segmented genome. And these variations result in antigenic changes in the viral surface glycoproteins that makes for viruses possible to escape the vaccination efforts and using the antibodies. So now let's take a look at the classification. We have three types already, A, B, and C. But out of these three, the only A is highly variable antigenically. And we blame type A in most cases of the epidemics. So, and that's because influenza A strains are known for many mammal species. They may happen in humans, aquatic birds, domestic birds, poultry, pigs, horses, seals, whatever. And while traveling, so they may be passed from one species to another one. And during this passaging or traveling, the virus usually mutates and undergo different types of antigenic variations. Then influenza type B is antigenically less variable and it may cause epidemics, but usually it causes seasonal epidemics. Influenza type C is antigenically very stable and may cause sporadic mild illnesses, but they are quite rare. Few words about the structure. You see this pleomorphic and middle size, and the genomes are segmented single-stranded RNA of negative polarity. And in A and B types, we have 8 segments, separate segments, and we have 7 in influenza C viruses. The first 13 nucleotides at each end of each genomic segment are conserved among all RNA segments. These sequences are important in viral transcription. So usually one segment codes for one single protein, but then it is known that two segments of influenza virus, A at least, codes for two proteins. So if we take a look at the segments, that's important that first 3 segments code for protein that are called PB1, PB2, and PA. Why it is important? Because all those proteins, see, they are shown as red dot, as a red dot. So they combine all together to make the RNA transcriptase complex. That is, in fact, the complex with polymerase activity. And that will be responsible for viral transcription and viral genome replication. See, these 3 distinct proteins combine together to make a transcriptional complex. Then hemagglutinin mediates the one segment codes for hemagglutinin that mediate the attachment to the cell. So this is for hemagglutinin is for attachment. Then segment 5, NP protein, and you can see it here. This is nucleoproteins, right? That cover the segments of the RNA, and they make the helical structure of the influenza virus capsid. And then we have 6 segment for neuraminidase spike. That is the surface glycoprotein that are polymerized and make the so-called spikes. That has enzymatic activity and important in the virus entry and release out of the cell. Then the segment 7 code for 2 proteins, M1 and M2. M1 is the so-called matrix protein. That is the one, right? Major component of the virion. And it lies inside, in between the envelope and the nucleocapsid, and it mediates the assembly of the virus. While M2 is a membrane protein. Do we have it here? No. M2 is a so-called membrane protein. It is incorporated into the lipid bilayer. Oh, sorry, M2, right? And it is called integral protein. So we don't call it surface glycoprotein because it is not exposed to the outside. So, and it is integral protein, but it is important in making ion channels. So that may help in elimination of the surface envelope when the virus is translocated from the endosome into the cytosol in entry. Then segment 8 also codes for 2 proteins, 2 non-structural proteins. So we think that these non-structural proteins are important in the translation because they inhibit the nuclear export of mRNAs, and maybe they are important also in the splicing of mRNA. So, okay, we mentioned this already. So a few words about the glycoproteins, hemagglutinin and neuraminidase. So they are inserted into the envelope and form spikes with a size up to 10 nanometers in length. So, and they are most important so-called protective or principal antigens on influenza virus. So, because they determine the host immunity against this virus. And also they are responsible for antigenic variations on influenza virus. And the antigenic variation or variability of the virus is the way how this virus may escape immune response. The thing is that if we treat the virus with the detergents, the surface glycoproteins will be removed and the virus will lose its infectivity. So because it will be unable to bind with the cell surface receptor. So that's why these viruses are considered to be less resistant in an environment to compare with the naked virus like poliovirus we studied already, or rotaviruses or some others. Here you can see the M1 protein shell better, and here you can see the microphotograph with the ribonucleoprotein that make the helix composed of nucleoproteins and viral RNA. So then, how we used to name and classify viruses. they are divided into two genera. One genus is influenza virus A and B, and second genus is influenza virus C. So the division into these three groups is based on the antigenic differences exhibited by viral internal proteins or polypeptides, mainly NP and matrix. So, my dears, these differences allow us to divide viruses into types. But then the differences is in hemagglutinin and neuraminidase, or vice versa. Hemagglutinin and neuraminidase allow us to divide them into subtypes. So A, B, C are types, and the particular strains of maybe of particular subtypes. So how do we name the viruses? It is based on the type ABC and from what host they had been isolated. But then, if the human is a host, we don't write it in the name. But if animal was a host, then we have to mention that it was swine, for example, or any other, or seal or whatever animal was. Then geographic origin, where the strain had been isolated. Then, of course, number, according to which it can be found in the register, and year of isolation, and antigenic description. For example, here this is swine virus, type A, then the host swine, then geographic origin, Iowa, where it had been isolated. Then the number of the strain, 15, then the year of isolation, 1930. And then the antigenic description. H is of the first type and N is of the first type. And then if you are talking about human isolate, then A, Hong Kong, geographical origin follows the type, so it means the human are hosts. Then number in the register, year of isolation, and Hong Kong is geographical name of the isolation. Then antigenic formula H3 and 2, and that is a human strain. Then I would like to pay some attention to genetic reassortment that is typical for viruses with segmented genomes. We mentioned it when we discussed rotaviruses, my dears, last time. And today we are talking about genetic reassortment in influenza virus. So you can see a cell co-infected with two different types of the viruses, one type and second type. But then when replication of the virus occurs in co-infected cell, then we may have the mixing of the parental gene segments. So, and the resulting virus may have segments out of both parental genomes of both types. So, and of course, they also may, as a result, have the glycoproteins that appear from one of the strains. So, then we have the dramatic changes if you compare this one and this one. So dramatic changes of the viral surface antigen structure. So, few words more about viral glycoproteins. Hemagglutinin can be of 15 types, from H1 to H15. But only 4 subtypes had been described to be isolated from humans. H1, H2, H3, and not so far ago, do you remember we said that avian flu appeared? So H5, that is the hemagglutinin typical for avian viruses. Now it can be isolated from humans. Then the function of hemagglutinin. It binds with the virion. It binds the virion to a susceptible cell. And the name is derived from the ability to agglutinate erythrocytes, because hemagglutinin binds with the surface of the erythrocytes. So, and this is the major antigen, antibodies against which may neutralize the virus. So, it gives rise to so-called neutralizing antibodies. And variability of hemagglutinin is responsible for biological evolution of the viruses in animal and human bodies, and in development of the subsequent influenza epidemics. Usually it has a quite complex structure as a trimeric molecule, and it consists of three intertwined hemagglutinin 1 and hemagglutinin 2 dimers. So, and each dimer has a larger globe and elongated stalk. The stalk is HA2 and the globe is HA1. And okay, on the globe we have an attachment site to bind with the cellular receptor, and F is a fusion peptide that allow virus to come out of the endosome when it enters, because it enters by receptor-mediated endocytosis. Then the second glycoprotein, that is neuraminidase. So we have altogether 9 subtypes of the neuraminidase from N1 to N5. And the known N1 and N2 subtypes are being isolated from humans, and the rest from animals. So neuraminidase is a sialidase enzyme. Why? What it does? It removes sialic acid from the infected cell, facilitating release of the virion from the infected cell when they bud of it. But also it prevents the aggregation of the viruses on the surface of the infected cell. Because if it stay aggregated, they are not active, not infectious. So, and at the beginning of the infection, the neuraminidase also helps virus to negotiate through the mucin layer to better reach the receptor in the respiratory tract. So it is also an important viral antigen, and also it is involved in the evolution and biological evolution of the virus in human being. Okay. So a few words about the antigenic changes in glycoproteins. They may occur by 2 ways. Antigenic drift. This is the one type, that's how we show it, an antigenic shift. Few words about it. Antigenic drift is a minor long-term changes that occur as a result of accumulation of point mutations in the gene. But then usually those strains will be selected in the human bodies, where these point mutations accumulation occurs in hemagglutinin or neuraminidase gene. So, and the changes in the genome result in the amino acid changes in the protein and the alteration of the antigenic sites of the molecule. So, if the antigenic changes occur, so then they escape recognition by antibodies by the host immune system. And usually when 2 or more mutations occur, we may see that new subtype appears in the population. But take a look here. It takes years, 10 to 15 years usually, for new subtype to appear by this way. But then we have another one that is called antigenic shift, that is associated with major antigenic changes. Do you remember you have a button shift on your computer, right? So then when you want to switch quickly from one thing to another one, so that is the mechanism of antigenic shift. So it is a genetic reassortment between two different influenza virus strains, for example, human and pig, or swine strain, or human and avian strain. And that occurs when one segmented genome of the virus reassort readily in the infected cell, co-infected with two different types of the influenza viruses. So then, as a result of this reassortment, a new subtype will appear within a year. See, this is one stage shift. Not many stages, many small stages here, but one stage shift. Or now we know that it may occur two-stage shift or three-stage shift in between different types of the viruses. Usually, antigenic shift doesn't occur in B and C viruses because the very few related viruses exist in animals. And that is the timeline showing influenza pandemics caused by influenza A. We mentioned a bit so, but then this H1N1 Spanish flu, 20 to 40 million deaths, no vaccines or drugs occurred that time. Then the Asian flu and Concon flu. One is H2N2, another one H3N2. These are new virus strains and they both give 1 million deaths. And there was vaccination already, but limited. Then cross-species transmission was shown in 2003 with avian flu with very high mortality rate of the outbreak. And then we have a new H1N1 strain that occurs as a result of the gene reassortment because 3 or 4 maternal strains that develops drug resistance. And then we have some outbreaks. For example, one of them is H7N9 with avian flu. That is how it is changing. So, I don't want to spend much time for virus replication. I just want to underline some very important features of replication that allow us to distinguish influenza virus from other RNA negative sense viruses. So really, the influenza virus replication has very distinguishing features. For example, that's typical for RNA virus to multiply or to replicate in the cytosol of the infected cell. But influenza virus transcription and replication occurs in the nucleus of the cell. Why it is so? Because when the mRNAs are transcribed from minor sense RNA fragments of the viral genome, then those fragments, they don't have cap. Do you know cap? Cap? This is a cap. See? When it happens, they don't have caps. So that's why this RNA fragment should be taken into the nucleus, and there the cap fragments will be cut off the cell, RNA, mRNAs, and ligated with the virus mRNAs. Only then they start to be possible to be recognized here on the ribosomes as mRNAs to start the protein synthesis. I hope you understand my Chinese language. Then the process of splicing occurs in viral replication. So, and splicing of mRNA may give possibility for two proteins to be produced out of one mRNA. You know that two segments of the viral genome may be spliced, viral genome RNA may be spliced. So two proteins will be produced out of one single segment. From one of them, M1 and M2 proteins, and from another one, non-structural 1 and 2 proteins. Then usually influenza virus multiplies very rapidly. And it is believed that new viruses are produced within 8 to 10 hours. That's why the usually incubation period of this virus infection is very short, from 6 to several hours, really several hours, that's possible. Usually the replication of the influenza virus is associated with the shutoff of the host cell protein synthesis, permitting selective translation of viral mRNAs. So briefly, virus attaches to the cell surface salicylic acids via hemagglutinin globe structure, and then penetration occurs due to receptor-mediated endocytosis, and then M2 protein mediates the acidification of the endosome and evacuation of the RNA segments out of the endosome, that is called uncoating. Then we have transportation of the RNA minus sense RNA segments into the nucleus, where the virus encoded polymerase, do you remember it is a complex of the PA protein and P1 and P2 proteins. So, and they arrange the polymerase that perform transcription of the minus sense fragments into positive sense RNAs. Then these positive capped RNAs are transported to the cytosol for virus protein translation. And then interestingly, some viral proteins will be transported back into the nucleus for the for the assembly of the nucleocapsids with newly replicated viral genomes. Then those proteins that are boxed will be transported into the nucleus. The rest will not be transported into the nucleus. The M proteins, as soon as they are produced, will be transported to modify the cellular membrane. Here, right? They will be here. The M protein will be transported here. And then the hemagglutinin and neuraminidase, while they will be translated, they will be glycosylated and modified in endoplasmic reticulum, and then by Golgi apparatus, they will be transported and expressed on the surface of the nuclear membrane. Cellular membrane, hemagglutinin and neuraminidase. Then the mature nucleocapsids will be transported towards the membrane modified with the M protein, because it mediates the interaction between surface glycoproteins and nucleoproteins, and then the process of bonding occurs. Then when the virus performs bonding, neuraminidase, see, the virus will stay attached to the acylic acid of the surface cell receptors. But then neuraminidase activity is to remove this virus off, release it, free it from the membrane of the infected cell. And that is very important because that is one of the targets for anti-influenza therapy. And then I won't pay so much attention at the symptoms. You all know symptoms of, I just want to mention the symptoms when we compare them with COVID symptoms, right? So this is a small clue, or you used to say hashtag, right? To differentiate between COVID and flu. First, these two have different cause factors. Flu occurs as a result of influenza virus strain infection, and COVID occurs as a result of, again, new strains of new coronavirus, COVID-2. What are the symptoms of flu? Usually very high fever and chills, cough, muscle or body aches, headache, and fatigue. So, most typically in COVID infections, we have cough, fever, shortness of breath, and fatigue. Prescriptions. In flu, usually oral administration of the antiviral drugs, very rare hospitalization, and the drugs can be used for both hospitalized or non-hospitalized patients. But then prescription in COVID is intravenously administered antiviral drugs, and they cannot be performed usually outside of the hospital settings. So the patient has to be hospitalized. Vaccination occurs against both types. So for the flu 3, there are multiple licensed influenza vaccines produced annually because of the antigenic variations. And we also have several COVID-19 vaccines that's supposed to be used. And if you are not vaccinated, you have to be vaccinated right now. Then recovery time, usually few days up to 2 weeks regarding the complications, because in flu, the most common complication is pneumonia caused by Staphylococcus aureus or some other very common, quite often normal floral pathogens like Staphylococci and Streptococci, while the recovery time for COVID here is written from 7 to 10 days. But you know that a lot of patients occur with so-called long COVID that have the consequences that may last for more than a year. Now, what do we have for laboratory diagnosis? First, we have rapid tests that may identify very quickly the influenza viruses. And these are fluorescent antibody tests, polymerase chain reaction, or I would like to add immunochromatography tests. I put it here, but it disappears somewhere. That are the most common. Immunochromatography, this is the most effective rapid test to identify influenza virus. Then apart from rapid tests, the influenza can be easily isolated, and usually we use chicken embryo for isolation, or quite often we may use now the special cell line is developed, continuous cell line, that is called MDCK, and this dog kidney cell line. And then when we isolate the virus, detection is performed by hemagglutination inhibition assay, hemagglutination and adsorption, and identification of the virus is performed by hemagglutination inhibition assay with a known panel of antibodies and direct fluorescent antibody assay also with the fluorescent monoclonal antibodies. Serology tests are based on the detection of antibodies to surface proteins and also the internal NP and matrix influenza virus proteins. So, and to find the antibodies in the patient serum, we use the same hemagglutination inhibition assay, but then the known antigen, laboratory strain of influenza will be used as a known antigen to identify a known antibody. So we do with the neutralization test and ELISA. Usually paired acute or convalescent sera are necessary. Normal individuals usually have influenza antibodies against different strains. Fourfold increase will help us to indicate the influenza infection. Now let's take a look at the potential targets for the therapy or prevention over the virus infection. So for prevention, the antibodies can be used to block the hemagglutinin and prevent the binding of the virus to the target cell receptors. So endocytosis occurs, and then M2 protein mediates the fusion and the release of mRNA. So this can be prevented by using amantadine remantadine that blocks the function of the M2 protein. Then some more targets. These are the using of neuraminidase as a target, and zanamivir and oseltamivir and some other drugs with the different trade names can be used to block the activity of neuraminidase, really to block the release of the virus, to block the free of the virus. And that also works very well, but it works at the last stages of virus replication. So of course it's better to block it at the very beginning than at the very end. So there are some more potential targets, but by now we don't have any good drugs that may act regarding that may act for the prevention of the influenza virus development of the disease. So the prevention and control is really difficult because every single year the vaccine formula should be updated, because every single year the research institutes, they monitor what are the potential epidemic strains in the human and animal population, because the vaccine should be produced before the season of flu infection starts. So that's why usually we use, as a vaccine, we usually use a cocktail that contain usually both one or several types of type A viruses that are called actual strains, isolated in the previous outbreak and during the inter-epidemic time. So, but then if we are talking about the types of vaccine, that is what antigens are used. And if we are talking about the type of vaccines, there are so many types. And the anti-flu vaccines exist of all each single type that we have here. We may have whole killed virus vaccines. Usually the virus is grown in embryonated eggs. We have subvirional vaccines or subunit vaccines when the virus is disrupted and the only surface antigen is collected. No, subvirion, okay, split the virus. And then surface antigen vaccines, subunit vaccines, when the only purified hemagglutinins are used. Then DNA experimental vaccines and the others. So, and every single year, we supposed to be vaccinated before we get flu naturally. And this year, this is called New Influenza Vaccine Recommendations for 21, 20, 21st, 22nd season. So, all seasonal influenza vaccines expected to be available for this season, and they are quadrivalent. And they usually contain, my dear, this is irrespectively on in which country they are produced. In USA, in Russian Federation, or Ukraine, or Great Britain, they all will be of same formulation. Because that is for worldwide spread of the flu. So they will contain hemagglutinins derived from one influenza A and H1N1 of particular strain virus, then one from H2N3 particular virus, and two hemagglutinins from B strains of the virus. Of two distinct lineage, Victoria and Yamagata lineage. And then please don't take the risk this season and get the flu vaccine that I did already. So, few words about the epidemiology. You already understood that our main headache is because of influenza A virus, because only this one undergoes genetic reassortment of hemagglutinins and neuraminidase. My dear, this is the picture from Nature Review Microbiology, and this is how the last 2009 strain of swine flu appeared. So first, the virus had been transported in nature in between shorebirds and waterfowl birds. Then they had passed to the domestic birds and then to swines. Why? Because in some countries, for example in China, because of the type of farming or breeding of animals. So if there's not enough space, enough space, the domestic birds or domestic animals communicate very often and keep tightly in the same area. So that's why the influenza virus has chance to recirculate there and back again in between birds and pigs. Usually pigs occur as mixing vessels, where a lot of strains of influenza virus may reassort. Then due to this reassortment, then the reassortation occurs between birds, pigs, and human strains, two types. So then the resulting pandemic strain occurs as a result of mixing of genes from the waterfowl, birds, pigs, and humans. That was a resulting strain. Why pigs serve as mixing vessels for assortments? Because their sialic acid receptors match with the hemagglutinins of many different types of subtypes of the hemagglutinin taken from different strains. So then, the thing is that influenza outbreaks occurs in waves, and the period between waves of influenza is usually 2 to 3 years. And the interepidemic period for type B is 6 to 3 years, so it takes longer. Every 10-40 years, pandemics usually occur, and they depend on the appearance of new subtype of influenza A, and you had the example of it, right? What do we need to control the influenza infections? Surveillance for influenza outbreaks is necessary. So it is required to identify the early appearance of new strains. And the aim is preparing vaccines against the new influenza strain before an epidemic season occurs. So the vaccine should be done available by September at least. And the surveillance is based on the isolation of many different types of the viruses with an altered hemagglutinin and or neuraminidase. And concerning avian influenza A virus with the formula H5N1, by mid-2008, 385 human cases of H5N1 influenza virus have been documented with 63% of mortality rate. So that is very important to predict whether any other avian the reassortments with avian flu occurs or not in the human population. And here you can see the control measures. So take a look at them and these control measures. And that's very important that the control measures should be performed regarding the most important source of the strains for reassortment. This is avian influenza viruses, because so a lot of poultry breeding plants occur everywhere, so that is very important to keep avian influenza viruses far from the human population and the bird and the breeding animals population. With this, we finish our lecture, my dears. See you next time with the lecture on, I think, or I should take a look at the plan. That might be hepatitis viruses. Goodbye, my dears. Take care, stay healthy, wear your masks, please. Make vaccination against coronavirus and against influenza. It is very important to prevent circulation of these viruses in the human population. And no circulation, no new strains appearing for both types. Goodbye, my dears.